Thank you for your introductions. Hello everyone, my name is Masato Kosuge and from the Hormonal Laboratories at Waseda University. Today I will talk about evolutionary arms races between transportable element and KRAB-ZFP families. In these studies, I reconstructed the histories of the battles between transportable element and human genes. First, let me explain about transportable elements TEs. TEs are mobile and parasitic DNA sequence within their host organism, host genomes. They can transpose through the mechanisms like copy and paste or cut and paste, allowing them to insert into the different regions of the genomes. Occasionally, these insert into the genes and can disrupt gene functions. Moreover, these constitute 46% of the human genome. Therefore, it is essential for the host organisms to evolve the suppression mechanism of TEs. Next, let's talk about KRAB-ZFPs. KRAB-ZFPs act as a transcription repressors for TEs in cooperation with various factors such as TRIM28, DNA methylation enzymes, and histone modification enzymes. Approximately 400 KRAB-ZFP genes are encoded in the human genomes, and this gene families has expanded to suppress TEs. Each KRAB-ZFP targets different T families, collectively covering a broad range of T families. It is hypothesized that an evolution arms race has occurred between Ts and KRAB-ZFPs. Previous studies have reported that KRAB-ZFPs co-emerged with their T targets, suggesting that KRAB-ZFPs emerged to suppress new Ts. Furthermore, one T families, line families, has been reported to evolve to evade from KRAB-ZFP suppressions. In other words, Ts and KRAB-ZFPs are engaged in a repeated cycles of suppressions and evasions, resuppressions, and evasions. However, the extent to which T families have undergone these arms races with KRAB-ZFPs remains unclear. Based on this background, we aim to uncover the histories of evolution arms races between T families and KRAB-ZFPs, and to debate the impact of Ts on the evolutions of their host organisms. This slide illustrates the designs of our studies. To uncover the histories of evolution arms races, we systematically conducted 3 types of analysis. Initially, we identified KRAB-ZFP targets through the reanalysis of large-scale chip-seq data for KRAB-ZFPs. Next, we compared the evolution ages of KRAB-ZFPs and Ts. Finally, we constructed a phylogenetic trees of T families. By integrating these analyses, we tried to reconstruct the dynamics of the evolution arms races between Ts and KRAB-ZFPs. First, we searched for the cases where T families have evaded from KRAB-ZFP suppressions as a signs of evolution arms races. We calculated the binding rates of KRAB-ZFPs to T subfamilies using chip-seq data and screened for the cases where the binding of KRAB-ZFPs was lost in younger T subfamilies compared with older subfamilies. This approach allowed us to identify the candidate for T evasion events from KRAB-ZFPs. This figure shows the result of our screening. We identified 60 candidates for T evasion events from KRAB-ZFPs. with many found in lines and ER buoys. Among them, L1P5' end, already known to undergo evolution arms race with KRAB-ZFP, was included on the top. These results suggest that several T families potentially have evaded from KRAB-ZFPs. In particular, we focus on LTR7/8, which is crucial for pluripotencies of human ES cells. LTR7/8 is a type of endogenous retrovirus that has evolved in the genomes for tens of millions of years. First, we examine the binding profile of 11 KRAB-ZFPs targeting LTR7/8. The left side of this figure shows phylogenetic trees of LTR7/8 ordered from older to younger copies. The right side of this figure shows a binding profile of KRAB-ZFPs. As seen in the figures, 5 KRAB-ZFPs targeted older copies, but did not target younger copies. In contrast, younger copies were complementarily targeted by other KRAB-ZFPs. This result suggests that turnovers of KRAB-ZFPs occurred during the evolutions of LTR7/8. Next, we examine the chronological relationship between the evolutionary ages of LTR7/8 and KRAB-ZFP. This figure shows the correspondence between evolutionary ages of KRAB-ZFPs and LTR7/8 subfamilies. Our analysis reveals that 3 KRAB-ZFPs indicated in blue colors co-emerged with the oldest subfamilies and targeted them. But these KRAB-ZFPs did not target middle-aged and younger subfamilies, suggesting that middle-aged and younger subfamilies have evaded from these KRAB-ZFPs. Additionally, the older KRAB-ZFPs alternatively targeted middle-aged subfamilies, and finally, ZF19s co-emerged with the youngest subfamilies and targeted them. These results indicate the chronological co-evolution dynamics between LTR7/8 and KRAB-ZFPs, suggesting that LTR7/8 have undergone the evolution arms race with KRAB-ZFPs. Finally, we explore the relationship between the evolution arms race in LTR7/8 and its regulations by KRAB-ZFPs in human ES cells, because LTR7/8 is highly upregulated in human ES cells and contribute to the pluripotencies. We used RNA-seq data from TRIM28 deficient human ES cell alternatively, because TRIM28 is essential for KRAB-ZFP suppressive function. We examined the alterations of the LTR7/8 expressions and the impact on genes near LTR7/8 copies. This figure shows the alteration of LTR7/8 expressions. In TRIM28 deficient human ES cells, older and middle-aged LTR7/8 subfamilies were specifically upregulated, but younger subfamilies were not. This result suggests that TRIM28 suppressed LTR7/8 families in a subfamily-specific manner in human ES cells. Lastly, we examine the expression changes of genes near LTR7/8 copies. This figure shows the proportions of genes downregulated or upregulated genes near each LTR7 subfamily. We observed that the genes near LTR7 copies tended to be upregulated in TRIM28 deficient human ES cells. This result suggests TRIM28 potentially suppressed nearby genes by LTR7 copies in human ES cells. To summarize, in this study, we systematically explored evolution arms races between T families and KRAB-ZFPs. We identified several candidates for T evasion events from KRAB-ZFPs and reconstructed the dynamics of evolution arms race between LTR7 and KRAB-ZFPs. Moreover, we observed TRIM28 potentially regulates the expressions of LTR7 and nearby genes in human ES cells. These results suggest the evolution arms race with KRAB-ZFPs have occurred in more broad range of T families and potentially co-opted in the gene regulatory networks. Finally, I would like to thank our collaborators and the members of Hamada Laboratories. And these studies were published 2 weeks ago. Please check it out. Thank you for your attention.
We have time for a question or two. If anyone would like to ask one, you can come up to the microphone. Yes, please.
Thank you very much. I didn't know this system, but it is very interesting. Thank you. I was just wondering if you said that TRIM28 regulate nearby genes. Isn't it possible that it is the transposable elements that regulate nearby genes?
Yes, previous studies have reported transposable elements act as enhancers or promoters of genes. So in this case, we think that LTR7 act as enhancer or promoters of nearby genes. Okay. Thank you.
Well, thank you. I think we'll have to move to our next speaker, but thank you again.
Thank you for your attention.